Hi everyone, this is Dr. O'Connor. In this video I'm going to talk about proteins and amino acids, and then I will do a second video on enzymes. I hear my neighbor's dog barking and there's really nothing I can do about it, so please excuse that. Hopefully they'll let him in pretty soon. Okay, let's go ahead and start. Let's define a protein. And it is a large biological molecule, and it's made of many amino acids that are linked together through what we call peptide bonds. These are actually amide bonds, and we'll take a look at that in a few minutes. An amino acid is a molecule that contains both an amino group, this NH2 group, and a carboxyl group. And there's also an R group, or a side chain, and these are all bonded to a central carbon atom. So for example, let's go ahead and scroll down here. Okay, so here we have an amino acid. Notice there's this carbon. It's bonded to the carboxyl group, the amino group, this R group. This is a variable group, and we'll talk about the R groups in a little bit. And this also has a hydrogen here. Okay, so these amino acids are called alpha amino acids, and these are amino acids in which the amino group is bonded to the alpha carbon, which is the carbon atom that is bonded to the carboxyl group. Okay, so this is the basic structure of all of the amino acids, our alpha amino acids that we're going to talk about here. We can divide the amino acids into 4 different groups. We have the nonpolar amino acids, and that means that the side chains are either aliphatic or aromatic, meaning they're hydrophobic, right? They're water-fearing, so they're nonpolar. Then we have the second type, the polar uncharged amino acids. And basically, these amino acids have side chains that contain functional groups that are polar. For example, maybe alcohols, an amide group, and so on. The acidic amino acids, what we have is a side chain that contains a carboxyl group. And we'll talk about those in a little bit. And then of course we have the basic amino acids, and in these amino acids the side chain will contain a nitrogen that can act as a base, so an amine nitrogen. These are called positively charged amino acids, and then the acidic amino acids are called negatively charged amino acids. So we'll take a look at that in a moment when we talk about zwitterions. So let's go on to our next slide here. This is a table from your book, and it contains all of the amino acids. All right, so it shows so for example, these are grouped. These are the nonpolar amino acids: alanine, isoleucine, leucine, phenylalanine, proline, tryptophan, valine. Okay, these are all the nonpolar ones. In other words, they have nonpolar side chains. Okay, the side chains are shown in red. So for example, alanine, it has a methyl group as a side chain, and that is nonpolar. Isoleucine, this is the side chain, a nonpolar side chain, and so on down the list. Here we also show the 3-letter abbreviations for these amino acids. And yes, you should be familiar with the names of these amino acids, and you should know by the time you're finished with this module, you should be able to differentiate between nonpolar, polar, acidic, and basic amino acids. Okay, so let's take a look here at the polar uncharged amino acids. And again, they're all listed here. Okay, we have cysteine, glutamine, glycine, threonine, tyrosine, and so on. Again, the 3-letter abbreviations for these amino acids. And let's go ahead and take a look at the side chains. So these side chains here, this one here, for example, it has this carbonyl group here and this amino group. Okay, so this is very polar. Here we have a side chain that contains this group here. Again, you've got this carbonyl group, and that is going to be polar. So these are all the polar side chains. So again, make sure that you know and that you can identify these polar and non-polar amino acids, okay? Because I will ask you on the exam. I'll probably give you a structure and I'll ask you, is this amino acid polar, non-polar, acidic, or basic, okay? Here we have the acidic amino acids, aspartic acid, glutamic acid, again, the 3-letter abbreviations, and as we can see here, in the side chain, we have the carboxyl groups on both of these, okay? And then, of course, our basic amino acids, we have 3 of them here. And again, we have the amino nitrogens, or the amine nitrogens, in each of the side chains, which makes them basic. So make sure that you look at that table and get to know it. So we're going to be talking about proteins in this chapter. And proteins, there are different classifications of proteins. And here's a table that I put together with the type of protein, the functions, and some examples. And so we have enzymes, which are proteins, and they act as catalysts. And one example would be amylase, and that is important in digestion of carbohydrates. Some proteins are hormones, and hormones regulate body functions by carrying messages to receptors. And insulin is a good example of a hormone that you're all familiar with. There are storage proteins, and myoglobin, that's a storage protein because it stores oxygen. We have transport proteins, and these carry substances through the aqueous body fluids. Actually, albumin, this is a transport protein, and it carries those hydrophobic fatty acids through the blood. We have structural proteins, and these provide mechanical shape and support. Collagen is a good example of a structural protein. Protective proteins, and these defend our body against foreign matter. And contractile proteins, these actually do mechanical work. So myosin and actin, these govern muscle movement. So we see the proteins there are different types, and they have many different functions, okay? It turns out that the alpha carbon of all of the amino acids is chiral. So these alpha carbons are chiral, except for glycine. And we just looked at glycine, and that's because glycine has 2 hydrogens bonded to that alpha carbon. All right, so we can draw these as Fisher projections, and this illustrates the D and L configurations of the amino acids. And we did that way back when we looked at carbohydrates. So there are some D amino acids that occur in nature, but only the L amino acids occur in proteins. And remember, the plus and minus signs, those indicate the direction of rotation of the plane polarized light by that particular amino acid, just like with the carbohydrates. Amino acids are what we call amphoteric, and remember back in Chem 251, we talked about amphoteric substances, and that means that the substance can react as an acid or a base. Water is a good example of an amphoteric substance. So alanine can react with a strong base here, or it can react with a strong acid. So in other words, in this case here, alanine is reacting with a base, so it is acting as an acid. My pen is not working right. And here, alanine is reacting with an acid, and so it is acting as a base. Amino acids exist in neutral solutions, okay, pH is 7, as zwitterions. Funny word. A zwitterion is a neutral, I should write that down, a neutral, if I can ever get it written here with this pen, it's a neutral dipolar ion, and this dipolar ion is formed by an internal transfer of a proton from the carboxyl group to the amino group. So we see what happens here. This hydrogen here, this acidic hydrogen, is transferred from this carboxyl group to this amino group through this lone pair of electrons. And what we end up with here is this zwitterion. And it is indeed neutral because we have one negative charge and one positive charge. And of course, those add up to 0. So obviously, these amino acids have acid-base properties. So the carboxyl group obviously is the acidic group, and the amino group is the basic group. And as I said before, these groups can undergo what we call intramolecular meaning within the molecule. So they can undergo intramolecular acid-base reaction. So again, as I said before, the proton from the carboxyl group is transferred to the amino group, which produces the zwitterion. Remember, the zwitterion has both a positive and a negative charge, and it is neutral. Okay, so because they are zwitterions, amino acids, they have many properties that are common for salts. So amino acids are crystalline, they have high melting points, and they are soluble in water. So in acidic media, meaning low pH, the zwitterions will accept a proton. So here's a zwitterion. So we have an acidic medium, so a lot of hydrogen ions or protons there, right? And it will accept a proton on the basic carboxylate group here, okay, and that'll leave a positive charge on this group here, okay, so that'll be positively charged. In basic media, where we have a higher pH, these zwitterions will lose a proton from this acidic part here, okay, and when this loses a proton, it becomes this ion here with a negative charge. So it's it's not neutral anymore, it's negatively charged. Up here, it's positively charged, okay? What does that mean? It means that the charge of an amino acid molecule at any time is going to be dependent on the pH of the solution. Now, we have something important called an isoelectric point. This is the pH at which an amino acid has equal numbers of positive and negative charges. And what that means is, if you have an equal number of positive and negative charges, the overall charge is going to be 0. So we can look up these isoelectric points of all the amino acids. So for example, here lysine, its isoelectric point is at a pH of 9.7. Glycine, 6.0. Aspartic acid, it's going to be a lower pH, 2.9. So the pH of aspartic acid at the isoelectric point is 2.9. It turns out the protein molecules can be separated in solution because of these positive and negative charges. And we use a method here called electrophoresis. And electrophoresis is movement of an electrically charged substance under the influence of an electrical field. So you'll have these two sides here, two electrodes. One is positive, one is negative. Okay, we'll expose a protein solution to this electrical field, and the movement of the molecules is going to be dependent on the pH of the solution, the strength of the electric field, and the charge of the particle. Also on the size and shape of the particle. The mobility of the protein, again, is going to depend on the pH of the medium. If the pH is at the isoelectric point, that means there's zero charge, the protein is not going to move. If the species is negatively charged, it'll move towards the positively charged plate. If it's positively charged, it'll move towards the negative charged plate. If it's, again, at the isoelectric point, it's not going to move to either side. So this is something that is used to check protein concentrations in the blood. So isoelectric points are found at pH values ranging from 7.8 to 10.8 for basic amino acids, 4.8 to 6.3 for neutral amino acids, 2.8 to 3.3 for acidic amino acids. So let's talk a little bit about peptide bonds, or peptide linkages as they're called. These are amide bonds. So what will happen is a carboxyl group of one amino acid reacts with the alpha amino group, the NH2 group, of a different amino acid. So for example, if we have two glycine amino acids, what will happen is peptide bond, okay, or this amide bond, will form between the carbonyl carbon of one amino acid and the nitrogen of the other, forming this amide group. But we call this a peptide linkage or a peptide bond. So in this case here, we would write the sequence of this dipeptide, if you will, using the three-letter abbreviation GLY and then a hyphen and GLY. Sometimes they won't use a hyphen, they would just write it like so, GLY, GLY, and that's to show that the two amino acids are linked together by a peptide bond. So this would be a dipeptide. There are two amino acids. If there are three amino acids linked, it would be a tripeptide. 4 would be a tetrapeptide, 5 a pentapeptide, and so on. Peptides containing up to 40 to 50 amino acid units are called polypeptides. We refer to the amino acids in a polypeptide or a protein as residues, okay, or amino acid residues. Longer chains of amino acids are called proteins. So when you have more than 50 amino acids linked together, we call those proteins. When we have a peptide, okay, we'll have a linear peptide. One end is going to have a free amino group, and the other end will have a free carboxyl group, okay? So we call the end of the polypeptide chain with the amino group, we call that the N-terminal residue, and the end with the free carboxyl group is called the C-terminal residue. So for this polypeptide here, the N-terminal residue is this one here, the alanine, and the C-terminal residue is on the right-hand side here, and that is the glycine in this case. So we number the sequence of amino acids starting with the N-terminal residue that's written on the left, and then we continue numbering 2, 3, 4, 5, 6, and we end with the C-terminal residue, which is on the right. If we have a segment of one of these polypeptides or proteins that's not specifically known, we see that they enclose that in parentheses. There are a number of small naturally occurring polypeptides that are very significant when it comes to biochemical function. we know of over 30 different peptides in the present day. Many of these substances serve as hormones or what we call nerve transmitters. So the amino acid sequence and the chain length give a polypeptide its biological effectiveness and specificity. And I'll tell you what that means in a second. Let's go to the table. Again, this is in your text, so you can take a look at this. Here we have the name of some of these peptides and their primary structure and their general biological functions. Here we have something called substance P. It has this primary structure and it's a pain-producing agent. Here we have several others. Here we have neuropeptide FF, and this is the peptide sequence here. This modulates pain sensations and so on. You don't have to know these. These are just shown as examples. For a protein to serve a specific biological function, it must have a defined shape. So we describe proteins on 4 different levels: a primary structure, a secondary structure, a tertiary structure, and a quaternary structure. We're going to go through each one of these now. Primary protein structure is simply the sequence in which the amino acids are linked by the peptide bonds. That's all it is. We look at this structure here, this representation, and we see that along the backbone of this protein there's a chain of alternating peptide bonds and alpha carbons. Think of the side chains as being substituents along the backbone, and again those are bonded to the alpha carbons. Here's an example of the primary structure of insulin. And we see that insulin is actually composed of 2 protein chains, and they're linked together by these disulfide bonds. Remember, we talked about that in the organic chemistry portion. Now, I will tell you, amino acids have to be connected in the correct order if the protein is going to function correctly. So that is very, very important. A change in just one amino acid will change the biological function of the protein. In other words, it will not function the way that it's supposed to. For example, in sickle cell anemia, one amino acid, just one, glutamate, is replaced by a valine. And think about how awful sickle cell anemia is. So it's just, again, one of the amino acids is replaced by another amino acid. So later on, we're going to talk about DNA, and DNA stores all of the information in order for proteins to be produced that have all of the amino acids lined up in the correct order. Now, so we know about the primary structure of proteins. Let's talk a little bit about the secondary structure of proteins. And we characterize that as a regular 3-dimensional structure held together by hydrogen bonding between the oxygen of the carbonyl group and a hydrogen of the amino groups in the polypeptide chain. Okay, so 2 examples of secondary structure. The first one is what we call the alpha helix, and that's where the chain coils into this helical structure, and its shape is held by hydrogen bonding, along the backbone. So we can see that in all 5 of these figures. So for example, we have hydrogen bonding here between a hydrogen that's bonded to the nitrogen on this part of the chain to the carbonyl oxygen on the other part of the chain. So these hydrogen bonds are very important in secondary structure. The other type of secondary structure is what we call the beta pleated sheet, okay? And so here we see that the chains are parallel to one another, and again, there's hydrogen bonding between the hydrogens that are bonded to the nitrogen on one chain and the oxygen of a carbonyl group, okay? And again, this forms a sheet-like structure. So the two types of secondary structure that are important are the alpha helix and the beta sheets, okay? And those shapes are the result of hydrogen bonding. That is the main interaction in secondary structure. The tertiary structure of a protein is the shape, the three-dimensional shape of the protein molecule, okay? So the three-dimensional conformation of the protein is held together by many different types of interactions between the amino acid side chains, and that can be hydrogen bonding, ionic bonding, disulfide bonding. So let's take a look at this. This is just a representation, the blue ribbon-like structure here. That'd be the primary protein structure, and here we see some secondary structure in the form of an alpha helix, okay? And here we see a pleated sheet, beta pleated sheet structure, okay? But notice in the tertiary structure now, this is the folding, okay? This is the shape, the three-dimensional shape. So for example, you have interactions by, one group in this part of the chain and another group that is many amino acids away. And so, for example, here we have a hydrogen bond between peptide groups, okay? That forms this loop here. Here we have these hydrophobic side chains that are forming a water-free pocket. Here we have hydrogen bonding between a side chain and a peptide group, okay? Here we have some more hydrophobic interactions, okay? Notice how that's forming the shape. Here we have disulfide bonds, okay, between the side chains, between a side chain on this part of the chain, okay, and then over here on this side. So that's forming that kind of loop, if you will. And then here we have what we call a salt bridge, okay? So this would be one of those ionic interactions that we talked about, okay? Now, the fourth type of structure is called a quaternary structure. And quaternary structure is not found in all proteins. That's only found in some of the more complex proteins. The quaternary structure is the result of two or more smaller proteins that are held together by interactions. So these types of proteins are, again, made of two or more smaller protein subunits, as they call them. The quaternary structure refers to the shape of the entire molecule, and it's determined by how the subunits are held together, and they're held together by non-covalent interactions like hydrogen bonding, ionic bonding, and some other interactions. Again, not all proteins have a quaternary structure. Hemoglobin, that's an example of a protein that has a quaternary structure. We refer to hemoglobin as a conjugated quaternary protein, And we see the representation here. It's composed of 4 polypeptide chains. 2 of them are alpha chains and 2 of them are beta chains. And they're held together by the interaction of hydrophobic groups and what we call heme groups. I'll show you what those are in a minute. So hemoglobin is an oxygen carrier in the red blood cells. In the lungs, oxygen bonds to iron. And each hemoglobin molecule can carry 4 oxygen molecules. So in red here, this red part, these are the heme units that are embedded in each of the protein chains. But the 4 protein chains are held together, again, by hydrophobic interactions. Okay? So anytime our cells need oxygen, the oxygen is released, and carbon dioxide is then picked up and carried back to the lungs, and this is oxygen transport. So hemoglobin is a transport protein, and it has a quaternary structure. Okay, so we have 2 main classes of proteins. I'll talk about them in a minute. For right now, I just want to show you some examples. We have some fibrous proteins, and they have a fibrous shape. So they serve as structural supports, and fibrous proteins are water insoluble. An example is alpha keratin, found in hair, fibrin found in silk, and collagen, which we find in many tissues. These are all fibrous proteins. Okay, collagen, for example, it forms what we call a triple helix. So there are 3 protein chains that coil together to form that helical structure. So it's very, very strong. So we were just talking about fibrous proteins. They're tough, insoluble proteins, and their protein chains form fibers or sheets. It's the secondary structure that is mainly responsible for fibrous proteins: wool, hair, fingernails, muscle fiber, all of those. They're composed of fibrous proteins, which we call alpha keratins. They're composed mainly of alpha helices, and these helices are twisted together to form even more structure. Spider webs are made of fibrin, and this is a fibrous protein that's composed of stacks of beta sheets. Silk also has this structure. The next type of protein is globular protein. So globular proteins are water-soluble proteins, and their chains fold in a very compact shape, and they fold in such a way as the hydrophilic groups are on the exterior. That's so they can travel through the aqueous body fluid. The structures are not as regular like the fibrous proteins are. We have the hydrophilic side chains. Again, they tend to be on the exterior of the protein because of the way the protein folds, and they can travel through the aqueous body fluids to where they are needed. And globular proteins represent tertiary structure, whereas fibrous proteins represent mainly secondary structure. And we already talked about some fibrous proteins. We have keratins, collagens, elastins, myosins, and fibrin. Let's take a look at some examples of globular proteins. Insulin, okay, that's a globular protein, and insulin is actually a hormone. We saw this primary structure of that. Ribonuclease, that's an enzyme that catalyzes RNA hydrolysis, or the breakdown of RNA. We have hemoglobin, which we talked about, albumins. Those are transport proteins. We talked about the hemoglobin having heme groups embedded in each of the 4 protein chains. And this is a heme molecule. And notice the iron here in the center. The iron is bonded to these 4 nitrogens. Myoglobin is a globular protein, and it also stores oxygen, but it stores it in muscles for use when there's an immediate energy need. It's the oxygen carrying portion that has the heme group, this one right here. and that's embedded again within the polypeptide chain. A conjugated protein is a protein that incorporates one or more non-amino acid units in its structure. And these non-amino acid units aid in the function of the protein. Chemical properties of proteins. Ignore I put that in there and I should have taken it out. Anyway, hydrolysis. Proteins undergo hydrolysis, but it's only the primary structure. It's those peptide bonds that are hydrolyzed to yield amino acids. Remember, those peptide bonds are amide bonds, okay? And we know that amide bonds are hydrolyzed. Well, it's the same with the proteins. The peptide bonds are hydrolyzed in order to yield the free amino acids. So really this is the reverse of protein formation. We're going to talk about protein digestion later, but most of it takes place in the stomach and small intestine. And all of these chemical reactions are catalyzed by enzymes, which are also proteins. We'll talk about enzymes in the next video. Individual amino acids, once digested, the individual amino acids are absorbed through the intestine wall, and then they're transported through the bloodstream to where they are needed. Let's talk a little bit about something called protein denaturation. And denaturation is a disruption in protein shape, but it does not affect the primary structure of the protein. The only way you can affect the primary structure of the protein is through hydrolysis of those peptide bonds. So let's say you have a protein here that is folded a certain way, and if it's exposed to heat, what'll happen is this structure will start to unfold. So in this case here, we see that the structure is unfolding from a globular shape to this randomly looped coil. When a protein is denatured, that's accompanied by changes in physical, chemical, biological properties. And there are 6 main agents that can cause denaturation of a protein. In other words, if a protein is denatured, it cannot function as it is supposed to function. Okay, think about when you cook something. You cook an egg or you cook a piece of meat. You are denaturing those proteins. You're not hydrolyzing those proteins. We have to digest the meat or the egg in order for hydrolysis of the primary structure to take place. But the heat will denature the protein. So why does that happen? Well, if you have a globular protein and you have attractions between the side chains. So if the temperature gets up to 50 or above, that's going to weaken those attractions between the side chains, therefore causing the protein to unfold. When you cook meat, it converts insoluble collagen into a soluble gelatin. Mechanical agitation or mixing. So for example, when you beat egg whites, what happens is you're denaturing the proteins in the egg whites. Okay. Detergents, soaps, that can disrupt association of the hydrophobic side chains. Remember, what do? We use soaps, right, to dissolve grease, which is hydrophobic. So we have those hydrophobic side chains, and we have those pockets that are made in the protein because of the interaction between the hydrophobic side chains. Put a little bit of detergent in there, that's going to disrupt that, and therefore the protein starts to unfold. Organic compounds, so some polar solvents like acetone and ethanol, they can interfere with the hydrogen bonding. So what they'll do is, if you add ethanol or a polar substance, they're going to compete for those bonding sites. And again, we know the secondary structure and tertiary structure is due to hydrogen bonding. If we change the pH by adding excess acid or hydroxide ions, what will happen is the hydrogen ions and hydroxide ions will react with acidic and basic side chains in the amino acids, and that will disrupt those salt bridges. So basically when milk turns sour, that's exactly what happens, and the proteins coagulate, if you will. And then we have inorganic salts. So if I add a salt, high concentration of salt, we have that those dissociate into ions, and those can also interact with the salt bridges, therefore causing unfolding of the protein. One thing that I should tell you is that we need to obtain protein from our diets because it is not stored in the body. Children, of course, need protein for growth, and adults need it to replace the protein that's lost each day in biochemical reactions. Now, it turns out that of the 20 amino acids, 10 of those amino acids are not synthesized by adults, and we must obtain them in the diet. So we have them here: arginine, histidine, isoleucine, leucine, lysine, methionine, phenylalanine, threonine, tryptophan, and valine. So we need to obtain these amino acids from our diet. So we talk about complete proteins. A complete protein contains all of the amino acids. So meats would be complete proteins. A vegetarian, they have to balance their protein. They have to make sure that they get all of the amino acids from their diet. So these amino acids that the body cannot synthesize are called essential amino acids. And again, essential amino acids must be obtained from the diet because the body cannot synthesize these amino acids to meet the nutritional requirements. Anyway, that's it for proteins and amino acids. You should read your text, and of course, but there is a whole section there on tests for proteins. I am not going to ask you about that. That's something you can do in the lab, but don't worry about that section on protein tests. Don't read it. Oh, you can if you want, but I am not going to ask you about that. That's probably something you're going to do in the lab, so I'll let your lab instructors take care of that. So have a good evening, and I will post a video on enzymes. I will do that in the morning. If you have any questions, I know this material is tough, so you need to come see me and ask those questions. Have a good evening. Bye-bye.